Hello, welcome to El Zoominario. Today we're very excited to have Nahui Medina Chavez. Nahui is originally from the state of Guerrero, Mexico. From a young age, she showed a keen interest in biology and health sciences. During her high school years, she had an opportunity to work as an apprentice in a laboratory at the Institute of Technology of UNAM under the guidance of Dr. Federico Sanchez. This experience was crucial in shaping her career choice. Subsequently, in 2008, she began her undergraduate studies in genomic biotechnology at the Autonomous University of Nuevo León. And 2 years later, she pursued her graduate studies at the same institution, ultimately earning her doctorate in science with a focus on biotechnology. Oh. During her doctoral research, she specialized in the culture and isolation of halophilic Archaea from Cuatro Ciénegas, Coahuila. Currently, she is in the third year of her postdoctoral research at the University of Minnesota, working at Mike Travisano's laboratory, where she is doing experimental evolution within the Archaea domain. Thank you very much, Nahui. We're very happy to have you here.
Thank you. Yeah, thank you. Well, hi all, nice to meet you. I'm super excited to be here because I've been waiting to give this a small talk for a long time. So the name of my talk is the Archaea, Earth Science, Earth Ancient Microbial Architects. So first, I'm going to start with one of the central questions in biology, and is that how to explain the remarkable biodiversity that Earth has, right? And because of that, biodiversity not only describes all forms of life, but also all levels of life, and it can go from individual genes to community of species. And it's also worth noting that biodiversity has sustained life on Earth for billions of years, and there are several known drivers that contribute to diversification. So, for example, in I don't know if you might remember, maybe for some of us, when I was in high school, I remember when they teach me how life was organized, it was organized in kingdoms, right? But back in the 70s, Carl Wood that I show here, thanks to using molecular markers like 16S, 18S, and ITS, and some others, was able to do phylogenetic reconstructions that proved that all life was held within three major groups and domains. And I'm talking about this because phylogenetic reconstructions is a way to measure in a way how big in the extension of the biodiversity in the planet. So if we remember a little bit of this basic concept of the domains, the three domains that we all know now, the eukarya, bacteria, and archaea. This later was, at the beginning, when we didn't know archaea was a different domain or a different group, we thought that basically the microorganisms that belong to that ones, which actually it was called before archaeobacteria, was thought to belong to bacteria domain, only containing basically microorganisms that were thriving in extreme conditions and were poorly sampled and poorly misunderstood. So what are the archaea, right? What is the archaea domain? I'm sure many of you have heard of it, especially lately. I can see that it's a trending now. So the archaea basically are prokaryotes of extreme nature, although we have known from the past decade that basically we can find them in mesophilic or normal conditions. They can be found, probably you can find some of them in the oral and the buccal cavities. We found some in our belly buttons. People that tend to sweat a lot, they usually have a lot of archaea in their skin. So they're basically not only found or limited to extreme conditions. However, most of them are. Their cell wall is constituted of pseudomorine, which is totally different from bacteria. Basically their form, the link, let's say the links or the it's kind of a stronger than the peptidoglycan that belongs to bacteria. And of course, Archaea share common features with bacteria in a eukaryote domain. So now that a little bit that I'm talking about our Archaea, there's something that has come up just recently. And that basically, this is a very interesting fact of Archaea, is that thanks to the advent of sequencing technologies and better protocol sampling and culture protocols, we have reunited more sequencing data, allowing the phylogenetic reconstructions to be or would have major resolution. This has created one of the main debates in biology, which is whether the eukaryotes originated from Archaea lineages. So, for example, there's one Superphyla Asgard that has been recognized by holding several phylums that are closest taxa to eukaryote domain. So the first one, like around 2016-17, the first one was Lokiarchaeota phylum, was proposed to be the most related lineage to eukarya. And then later it was Hymdalarchaeota, and then just lately Hodarchaeota phylum. The debate, of course, is standing, and I'm sure as time goes by, we will continue finding new lineages that might help us to link and close that gap. So something that it's very important to recognize is that Mexico, now talking about a little bit more about biodiversity, is that Mexico has astonishing biodiversity, having several habitats, which means more species. And it's known that between 10 and 12% of the world's species are in Mexico. So in order to study early evolution of life without relying on fossil records, we found in Mexico an ideal place, and that is called Cuatro Ciénegas Basin. One of the features that makes Cuatro Ciénegas Basin in a polyextremophilic environment is that they have unusual and extreme stoichiometric ratios, especially on phosphorus. There's a lack of phosphorus. There's a scarcity of phosphorus. It's also known because there's been 3 decades already of research there. There's high microbial diversity, even though the place is very oligotrophic, and it's unexplored for many taxa. So this extreme physical-chemical conditions makes Cuatro Ciénegas Basin an ideal place to study, especially Archaea. So just recently in 2018, there was a paper that just came out from the main group that UNAM basically works within, and it was proposed that Cuatro Ciénegas Basin is an evolutionary bubble where all lineages basically evolve isolate, and due to the geographic isolation in this place. So Cuatro Ciénegas Basin is basically located at the northern side of Mexico and within the Chihuahuan Desert, and it's very particular the shape that it has. In Mexico, we call it as the butterfly shape. So this is basically where I used to work, where I retrieved many of the Archaea. So in March of 2016, basically after unusual precipitations, me and another researchers that we were there trying to look for places where we could retrieve some Archaea. We came up with this dome-shaped structures not far from the very famous Poza Azul, which is a blue pond, very well known in Mexico. And these structures were thriving in a shallow hypersaline pond. So if we do a closer examination, it revealed a finely stratified structure, which is basically a microbial mat. And it has this green, purple, and red layers overlaying a dark, deep stratum. So after some analysis, it was discovered that the gases within the dome mass were rich in methane and sulfur. So that reminds us a little bit of a replicate of the Archaean. So due to that, we decided to name it Archaeandoms. So we decided we decided to choose this particular place, the Archaeandoms, what we call Archaeandoms, and we decided to check what was the biodiversity in that specific place. So we took 3 samples in different times. Basically, we chose 3 temporals. It was in the first one in 2016, the drying temporal, then the wet, and then the dry. And basically, we took the same bubble just to try to correlate our results. So once we try to do the metagenomic analysis, we also grab that microbial mat and try to use it as a sample for culture in Archaea. So what we found, and we were very happy to find, is that we discovered the presence of Archaea domain, comprising basically around 5% of the relative abundance. And most this 5% is comprised by Euarchaeota, Crinarchaeota, Taumarchaeota, Nanoarchaeota, and Coarchaeota, which is basically, we never thought we were finding something like Coarchaeota, because basically most of them have been found in mines. And this is not a place where there are mines or conditions that Coarchaeota usually thrive in. So we also find that if we compare sites that have similar physical-chemical conditions to this one, and also trying to do a little bit of comparison with same metagenomic analysis, we find that the Archaeandoms held the majority in terms of Otus, the major number of species. So basically we find 230. However, we have continued over time. Right now we have 30 metagenomes, although that's been part of other PhD students, because basically this is what I'm showing you right now it was part of my PhD research. So with that, at that moment, it was probably by when we find this, it was probably around 2019, and we didn't find any other place, even Guerrero, which is very salty, right? It didn't have that many Archaea species. And once we start cultivating, we start getting new isolates, of course. And I can tell you it was a pain, because some of the colonies, and you might not believe me, but it's true, we find it after a year. And one of the issues we encountered is what I'm going to show you next. And it's basically once we start, I thought at the beginning that, okay, these plates are getting dry, they're not going to work. And at some point, I guess in my desperation, I forgot some plates in the incubator. And then months later, I find out that they actually are able to grow in the precipitated salt. It takes longer, of course, but they're very happy. And I still have some of these old plates, and you might think the bugs are dead, right? But they still thrive, and actually I can take some samples from that salty colonies, or this part of a precipitated salt with some colonies, put it into culture media, and they still grow. So I find out that basically that's also one of the best protocols for cryopreserving the microbes. So this is just a picture of the colonies and the growth on top of the salts. So what I'm doing next, I'm going to show you what I'm doing right now in my postdoctoral research. I'm continue working with Archaea. And when I was in my PhD, I used to have this Alobacterium salinarium that basically started to grow in 2016, and without knowing. I didn't know I was basically doing a laboratory evolution of Alobacterium salinarium, because I continuously transfer just to have it as a control of an Archaea over 4 to 5 years. And this is just a graphic that I'm showing you just to show you 4 years, but I'm still continuing the growth of Alobacterium salinarium. So with that, we decided to trying to simulate the original work of the long-term evolutionary experiments by Richard Lenski and Michael Travisano. So we start culturing this Allobacterium Selinarium along with the bacteria, that is Salinibacter ruber, just to have both domains. Especially because Salinibacter ruber is is an allophilic bacteria, and it can grow in the same amount of salt as the Archaea. So we pick basically 10 individual colonies as replicates, and then put them together as well in what we call a mix. So basically we have 10 populations of the Archaea growing separately, 10 populations of the bacteria, and then we have 10 tubes more where we have the mix of both. These 30 tubes basically have no selection. It's a batch transfer, but besides that, we are having these other 30 tubes with the same 10 Archaea, 10 bacteria, and 10 mix, where we're trying to put a selection pressure, and that's settling selection. We're trying to select for either big or those ones that might get into clusters and have a bigger or more complexity. So what we do basically is that each single tube, we take 1 ml, we put it into an Eppendorf tube. We wait around 5 minutes and let it settle in the bench. And we're trying to take out whatever it's in the bottom, 100 microliters, and then transfer it to a tube with fresh media. And with that, we have continued over right now 14 months. So basically in July, we turn a year of the experiment. What I can tell you right now, because this is something that we haven't published, we're basically in trying to get the first paper of this long-term evolutionary experiment. What I can tell you right now is that basically we saw rapid evolution because 10 transfer later, which was around 3 months, we start seeing a fast growth in both Archaea and bacteria. The mix behaves differently. Now, basically we transfer every 3 days. And well, we're continue doing this experiment, either settling and not settling. And right now we're planning to continue the experiment, but we are also in our way to do transcriptomic analysis as a way to measure the rapid evolution and changes within the generations, the populations, and both strains, of course. And that also, the mix can help us to understand a little bit of the relationship that have bacteria and Archaea. As we all know, as a way, let's say, to understand a little bit the relationship and interaction, the dynamics that both domains can have, while let's say we're doing this as a way to understand eukaryogenesis, especially because these bugs are extremophilic, and we all know that eukaryogenesis, one of the most accepted theories is that there was a syntrophy going on. It was an Archaea and then a bacteria. So we'll see. We're not sure right now what we're going to do with the mix, but we're excited to see the transcriptomics results soon. And with that, I don't have too much results to show because we're basically in the analysis phase right now. But what I can tell you is that if you want to read a little bit more about what we're doing, either with the Quaternions basing biodiversity, Archaea biodiversity, you can read one of the papers that just come out in August. This is in astrobiology, and one that I just wrote about. It has more results about the Archaea and Quaternions basing that we're using in the experimental evolution. And with that, I just want to thank to the CCB team in Mexico, Valeria Sousa from UNAM, Susana de la Torre in the University Autonomous of Nuevo León, and Abigail Torres, which was basically a huge help for me in the PhD. And my new lab, Travisano lab, of course. Mike Travisano, Maria Columbakidis, which is a PhD student, has helped me a lot with experimental evolution, along with an undergrad Chavez method. And with that, I will happy to take any questions.
Okay, I'm gonna stop the recording.